From Microbe TV, this is TWiP, This Week in Parasitism, episode 246, recorded on the 18th of October, 2024. I'm Vincent Racaniello, and you're listening to the podcast about parasites. Joining me today from New York, Daniel Griffin.
Hello, everyone.
What you got on your tie, Daniel?
So it is, so you know what it is it's a Friday, right? I was feeling bad. You know, when we record on Friday, we only get sexually transmitted infections. So I've got a purple spirochete on there.
Oh, my.
Right. So syphilis. We've got
Purple
syphilis, which
spirochete.
I want to point out, we have more syphilis right now in the United States than ever in history.
Really?
All right, I want to pursue that. But why don't you have Trichinella on your tie?
You know, I wore that yesterday. Was it yesterday?
Yeah, it's yesterday. Yeah.
One day
yesterday
early.
I had the nurse cell, and Dickson, who has not been introduced yet, so he can't talk apparently.
That's
But
right.
Maybe we should wait till he gets joined, and then I'm telling about what I wore yesterday.
Yes. Also joining us from Fort Lee, New Jersey, Dickson Despommier.
Hey, everybody. How you doing? It's a great day out there, by the way. It's really one of the wonderful fall days of the year. And temperature-wise, humidity-wise. And I'm all set to go trout fishing tomorrow and the day after that. I picked those days on purpose because they came into register with the weather. At any rate,
All right,
but
Daniel.
I'm so excited about today's program.
Yes, before that, though, Daniel, you were going to say something.
Oh, I was going to mention yesterday, Dickson, when I was recording This Week in Virology Clinical Update, I was wearing a bow tie that had a nurse cell on it. And, you know, and what it is, so it's the nurse cell, I think that's on our TWiP little logo thing, but it's repetitive pattern. So it covers the whole bow tie and there's like a nice white, you know, sort of way across it in the center. But this I found very disturbing. So I go into the ICU, right? And people, oh, what is that on your bow tie today? And I point out, I say, it's not a doctor cell, it's a nurse cell. Just silence. And everyone's like looking at me. I'm like, a nurse cell, Trichinella. Still,
and they still were silent.
What has happened to modern medical education, Dickson?
Well, it's gone down the tubes as well as you know.
So, Daniel and I were reminiscing yesterday.
Yes.
There used to be at least one course for medical students at Columbia in parasitology, right?
I think there still is.
Oh, yeah?
I think it's a first-year course, however.
It's sort of a truncated little,
It's a concession to the fact that New York City is a collection of tropical islands all brought together by commerce and, family ties and things of that sort. But we see it all, don't we?
But
you
of course,
still
really,
see it.
yeah. I say it's the center of the developing world because people come from everywhere. I do a lecture in Glasgow where it's all about like this person came from, this area, came from,
New York is the most connected city in the world.
I think that's true.
There's no other city comes close. People from everywhere come here, so we have all those microbes.
And restaurants.
So, Daniel, why is syphilis so high right now?
Yeah.
I worry about several factors. One, it's always education and awareness. Like people don't realize that it's an issue, and part of the tragedy there is the mother to child congenital syphilis.
Oh dear.
There also is, and people are aware. If this is a family show, but there's a lot less barrier protection being used during physical intimacy. And that there's several factors. I mean, some of them are good. I mean, the fact that we have pre-exposure prophylaxis for HIV, the fact that there's birth control, so there isn't that, which has been around for decades. But yeah, I think a lot of it is a lack of awareness and a lack of barrier contraception, and also if you don't make the diagnosis, you don't treat people, then you have these sexual chains of transmission, and that's a huge problem.
Is it treatable?
Very much treatable. Actually, a lot of, depending on the stage, you might use just an injectable penicillin, maybe one shot, maybe 3 shots, a week spaced apart. Or in some cases, you could even use, other regimens, except, in
Yeah.
pregnancy. Sometimes you actually have to do a 2-week course of ibupenicillin for, CNS during pregnancy. So
I remember when I was first starting out at Columbia as a technician and sort of eavesdropping in on the conversations of the doctors as they were reminiscing about their own past and the future of where medicine is going in New York. And they lamented the fact that it's all about funding for surveillance. At least those days it was. This was in the early 60s. And they said that if you surveyed for tuberculosis, for instance, which is a totally different way of surveilling than it is for syphilis and contacts, then the money that was shifted from syphilis to tuberculosis went down, but syphilis went up. Then they would shift the money back to syphilis, and tuberculosis would go up and syphilis would go down. So I don't know if that's still the case, Daniel, but we sort of put our money where other people's mouths are. And they keeps yelling and screaming, how come we're not supporting this? And the next thing you know, we're supporting it, but you have to drop something in order to support something else. And I'm afraid that's the way our economics has gone. Is that still the case?
It's what's the quotation? Violence is never the answer. Money is. But no, when we had Ben Lebrot of Floating Doctors, we were talking about this massive effort to control the screwworm and just how expensive it is to not do it. But then when you stop doing it, you don't keep doing it because, it's not a problem. Why are we doing this? You stop doing it, then it is a problem. So it's hard to have that momentum because a lot of diseases in sub-Saharan Africa are a victim of their own success.
Right.
When the incidence goes down, people are like, what are you even talking about? I don't even know what that disease is. Why are we funding? Let's wait till it's a problem.
African sleeping sickness is a good example of that, by the way.
That's it. Unfortunately, yeah. And who's going to be around to even be expert, right? How can you have clinical expertise in a disease if you don't see it? And so we need mechanisms for maintaining the expertise in these areas. Otherwise, we have to let it become a problem once again.
Well, you know that we have tried in the past. We have a series of lectures based on the 7th edition of Parasitic Diseases. We have a new edition coming out soon. I presume within the next 6 months. And it's updated. Whether people read it or not, that's another thing, of course. Maybe Vincent and I should revisit the lecture series and do some concentrated combination lectures on multiple diseases that are either vector-borne or food-borne or airborne.
Yeah, no, it might be worth circling back and doing, because I know we're sort of preaching here to the choir because a lot of our listeners are the people that are interested.
That's right.
Making sure they keep themselves educated on this. I actually got an email just yesterday from the Sudanese contact. Remember, we sent all those books to.
Oh, sure.
Yeah, and then there was the war, and who knows what exactly is going on. The university is having trouble to the point where he actually had to use the student's email to reach out to me. But no, I mean, a lot of parts of the world, what we're talking about is really critical. So.
That's right.
And these resources that Dickson has mentioned are free.
Yeah,
they are free.
They are free, and they're funded by our listeners, which
you bet.
Tremendous.
And the demand is there. I mean, every time we offer books for a class or for a school, all the hands go up.
Yeah.
So we know that the need is still there.
Yeah. And again, we need your support. But,
we do.
But we have some pretty exciting stuff to talk about today.
We
do. Even though perhaps the modern medical students are not up to speed. But I was trying to remind them, to give them that personal connection, that some of the stuff we're going to talk about here, there recently was a case where it was actually a friend of one of the PAs in that ICU who got sick, who was actually seen in our office with this malady. So maybe, Dickson, what are we talking about today?
Well, we're talking about my favorite worm. Actually, we're talking about something that I devoted, the first time I encountered this organism was in a class in 1960. II
Wow.
at Columbia University as I was going for my master's degree in medical parasitology. And the class was called Parasite Life Cycles. And there were a number of parasites that they had available in the laboratory. And they would display them for us and show us the pathological aspects and how easy or not so easy it is to handle them. Of course, we didn't have malaria, although we did have an insectary at the time. We were not raising the mosquito that transmits it. We were raising Aedes aegypti instead because it's much easier to raise, obviously. And a lot of diseases are transmitted by Aedes aegypti as well. Everybody knows that. It's also referred to as yellow fever mosquito. But so when it came to this organism, which immediately attracted my attention, the guy who was running the course, his name was Al Urinsky. He had just gotten his degree from the University of North Carolina from Dr. John Larsch, who was at that point a recognized expert on this particular entity. And so Al was very, very savant in how to handle it and how dangerous or not it was and how easy it was to maintain, which is all true, and how much we didn't know about it, even though it was discovered in 1835 in a remarkable series of events which took place in England involving the head of the British Museum of Natural History, Richard Owen, who ended up naming the organism, which in this case was named Trichinella spiralis. And they had to change the name because a search of the literature, which is pretty sparse in those days, but nonetheless, there was another organism called Trichinella.
Yeah.
So they had to call it Trichinella, little Tricha.
Oh, wow.
And so they ended up with that name. And for many, 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 many years afterwards, everyone thought that there was only one species of Trichinella, and that was Trichinella spiralis, the one that affects both animals and humans. In fact, it's almost as versatile a parasite as Toxoplasma gondii, and it's transmitted in exactly the same way, by the ingestion of raw or undercooked meat. And so any mammal throughout the world, unless you consider the insectivora, that's echidnas and platypus and several other egg-laying mammals, those are probably not susceptible to this infection, but they would be the only exceptions that I can think of. And I don't even know if it was tried or not.
Question. Like, could they get infected if, given exposure?
How could they get infected? Exactly.
Yeah.
An echidna eats ants. It's essentially an ant eater and a worm eater. And a platypus is a aquatic insect eater. So none of those two animals feeds on carrion, which is an essential ingredient. And in fact, one of the papers that we'll review today discusses the decline in the transmission cycles of animals that die a natural or perhaps not a natural death from a plague of some sort. And then the bodies are consumed by other animals which take advantage of these events. But we'll get to that. So the organism that I studied since 1971 until 1997, when my grant finally terminated, was called Trichinella spiralis. And that, for me, that was, I still think about it every day. I know, Vincent, you're totally committed to viruses and everything viral, and I'm committed to everything spiral.
Yeah.
So perhaps we've got something in common. And in fact, Trichinella has been shown to be a vector for a virus infection called LCM virus. And they've given it, they've actually shown that larvae derived from an animal with LCM can then transfer that viral infection to another animal through carnivorism.
Dickson.
Yes.
Maybe you could give us a brief overview of the life cycle?
I would love to do that. I mean, I just revel in this because the more I learned about this organism, the more I was attracted to the fact that it was a master of all trades, and it was really, truly a worm in a hurry. This is called a worm in a hurry. I'll give you the thumbnail sketch first. There are two phases to this life cycle. The first phase takes place in the small intestine of an animal that eats meat or muscle tissue, not brain, not liver, not kidney, not even heart muscle tissue. They have to eat striated skeletal muscle tissue, which harbors the infectious stage of the infection. And we'll talk about that too, because how can a dead animal with a worm that's dependent upon a living animal for its life serve as a conduit for the spread of an infection which depends on scavenging? And not scavenging among, let's say, birds or worms or other known entities that take care of carrion, but other mammals that actually take advantage of that as well. And meat is an incredibly valuable resource in nature. Its high caloric value is appreciated by virtually every trophic level. And meat, if one bite of meat is equivalent to 10 bites of vegetable material. So that's how much the caloric value is. And you don't have to sit around and munch away all day long like some grazers do in order to obtain your nutrition.
They don't have anything else to do, Dickson.
Well, they have one other thing to do, and two other things. They have to reproduce, and they also have to make sure that they're not eaten.
Yeah.
And having taken several trips now to Africa recently, I have come to deeply appreciate the fact that everything gets eaten eventually. Everything. We even witnessed the takedown of a baby elephant trying to cross the Zambezi River. And the mother was ahead of the baby. And when the mother looked around, the baby was gone. And the baby we all saw it. And then our attention was diverted by the herd of elephants crossing the river. When we turned around again, the only elephant we saw was the mother, not the baby. Then there was this enormous commotion behind the mother of the baby struggling to get away from the crocodile. And of course, it was in the jaws of the crocodile. The mother went over to try to help, to no avail, and she eventually gave up. And a little baby elephant was consumed by not just one crocodile, but I'm sure many different crocodiles, which brings me to a point. We now have discovered one of the species of Trichinella. Its name is Trichinella zimbabwensis. And you'll never guess what animal it infects, and it's the crocodile. Because its body temperature is around 37 degrees anyway, because that's the outside temperature. Okay, so let's see, where was I?
So
okay,
that's making me think that maybe crocodiles eat other crocodiles?
They do. Oh, no, they do. They absolutely do. That's the reason why a mother crocodile, well, we're going to drift off into various topics here, but
I think this is interesting with Trichinella, because some of the Trichinella seem to be somewhat species specific, right? There's a certain amount, right?
Yeah, you could say that, but in fact, I don't know of any human case. We don't know of any human case of Trichinella zimbabwensis. We don't know of any at all. But Trichinella pseudospiralis, Trichinella bertovii, Trichinella nelsoni, Trichinella spiralis, of course, Trichinella nativa, which is one of the ones we'll discuss today. We can all become infected by that. And in fact, nativa has been responsible for exterminating entire villages of people living above the Arctic Circle, whose only source of nutrition is meat. There are no gardens up there raising carrots and other vegetables, so they eat meat. And I was told, I'm not sure if this is true or not, that the word Eskimo in their own language means raw meat eater. And so a lot of Eastern Shore villages in Canada have been essentially, I was going to say decimated, but that's only 1 in 10. This is everybody gets a little piece of meat and nobody cooks it. And as a result, because there's nothing to cook with, basically. And everybody gets sick and everybody dies. It hasn't happened recently because of modern technologies. You actually can sell refrigerators to people living above the Arctic Circle so that they don't have to rely on nature for their source of nutrition directly from their hunting. But whenever they do, they're taking a chance. That includes animals like walrus. Walrus has actually been shown to be one of the animals that can transmit Trichinella nativa from person to person. And in fact, a very famous expedition back in the 19th century, Andre from, I'm going to say Sweden, but I think he was actually from Norway, and 2 other people in a hot air, in a hydrogen balloon, attempted to cross over the North Pole, to be the first people to cross over the North Pole. And they found their bodies something like 10 years later, and they had cameras with them, so they took pictures of everything they ran across during that time. They had enough supplies to last a little while. They had guns, so they were able to hunt. And so they hunted. And what did they kill? Whatever was threatening them, and in this case it was a polar bear. And the polar bear was consumed without cooking. And all 3 men ended up dying from trichinella. And the symptoms were meticulously recorded by the person who died last, who was Andre, because he was the last person to eat.
Wow.
The first 2 guys would eat first. He was the leader. He waited another day, and it delayed it just enough time for him to record the symptoms. And unfortunately, all the bodies were found, all the cameras, all the guns, all the evidence.
Is it so lethal typically?
No, it is not so typically. But if that's all you've got to eat, the answer is, yeah, it is. The trichinella doesn't want to kill you. It just wants to distribute itself throughout the environment. And so that's why scavenging is such an important life cycle part. Let me get back to the life cycle, though. He asked me about the life cycle. Okay. So part of the life cycle takes place in the small intestine, and the rest of the life cycle takes place in the muscle tissue of the same host. So the parasite doesn't actually complete the life cycle in the same host. It begins by ingesting larvae in the muscle tissue of an already infected host. The larvae are released from their muscle tissue by enzymatic digestion in the stomach and small intestine. The larvae then, immediately knowing where they are, penetrate into a row of columnar cells in the small intestine. And there, over a period of 34 hours, molt 4 times. That's what I said. This is a worm in a hurry. We don't know of any other nematode that molts to an adult within almost 1 day. And not even Caenorhabditis elegans does it that fast. Trichinella is definitely in a hurry. And the reason why it's in a hurry is because the immune system is running neck and neck right behind it, trying to catch up to it. And eventually it does, but not before Trichinella ends up producing live larvae as an adult worm in the small intestine. The larvae then penetrate into the villus from the columnar cells, and they either enter a lacteal, which is a lymphatic vessel,
so there's actually a sexual stage, right, in the small intestine?
There is a sexual stage. Yeah, there are males and females. Mating occurs. That's a little mysterious because we're not sure where it occurs. Does it occur in the lumen of the small intestine? Does it occur within the cells of the columnars? It's hard to imagine this, and we did some experiments to try to find out, but it was preliminary, and my grant actually didn't cover that topic, so I had to drop it in order to get back on track. But the point is that eventually, within 5 days after mating, the female worms begin to shed live larvae. Now, these larvae are much smaller than the ones that end up in the muscle tissue. They're only about 7 microns in length and 2 microns in width. They're almost like the size of a red cell, basically. But they've got a brain. They have nervous tissue. They have muscle tissue. They have stored proteins, which they will use immediately upon entering a cell. And when I say a cell, because they can enter any cell, they once they're in the circulation, when they reach a capillary and they get stuck, they then are, that's called a positive thigmotactic response. The pressure of the vessel around the worm triggers the worm to actually exit from the vessel and try to penetrate whatever cell they're near. If it's a muscle cell, of course, a striated muscle cell, then the parasite has found nirvana, and it begins its second phase, which is called the parenteral phase. The first phase is called the enteral phase. So the parenteral phase begins when the larva succeeds in finding a striated skeletal muscle cell. It then secretes stuff into the milieu of the muscle, and eventually all of those secretions of this worm end up reprogramming the cell. I know that sounds like a virus, and indeed I wrote an article once called Trichinella spiralis, the worm that would be virus. And it's because it uses metabolic messages that alter normal skeletal muscle behavior. And it ends up with, well, why don't I just show you what it ends up as? I happen to have
it. You have it there on your mug.
Right next to me. It ends up looking like that. Now, I don't care who's watching this show. Everyone knows that that is not a striated skeletal muscle cell. But at one point, it was. And that's what I spent 30 some odd years pondering. How does a striated skeletal muscle cell, fully committed to contracting and making work possible, get altered by an organism who has absolutely no use whatsoever for that cell, but has use for the tools which allows it to reprogram this cell? And if I could in some way put my hands around this nurse cell here, let's pretend this is the nurse cell, then this is the host circulation, which is elicited by the parasite through an alteration in the human genome to produce VEGF. And VEGF is a angiogenic factor, which then allows the vessel to seek out and surround this damaged portion of muscle. And the worm in 20 days achieves maximum size. If it's ingested before 14 days, it's not infectious. If it's ingested 15 days after infection in the muscle tissue, it's infectious.
But in people, it's a dead end because we don't eat people, right?
Well, most of us don't.
I was thinking about those 3 hot air ballooners up there in the Arctic. Could they have been consumed by something? And then they're not a dead end host at that point.
Listen, soccer teams that got stranded, the Donner Pass people. There's a lot of people who like people so much that they eat them. So
I think Alfred Packer was acquitted, by the way, just talking about the Donner Pass story.
All right. Spreading bad rumors. I don't want to do that.
Bad rumors about Alfred Packer.
I don't want to do that. What I want to say, though, is that for however long the host lives, that's how long some of the larvae from that infection can stay alive.
I mean, historically, human beings were consumed, unfortunately, by, carnivores, lions,
No,
crocodiles.
that's still being done, by the way. If you go to Tibet, when somebody dies in one of the religious groups there, they chop the body up.
Yeah, the
and put it
Tibetan
on a
sky
platform,
burial. Yeah.
and the condors come down and they eat it. But the birds will not get infected. Their body temperature is too high.
So do the nurse cells last the lifetime of the host?
I was going to say, some do. Not all. Some almost immediately become calcified, and then some will go on to live a year, 2 years, 20 years, 50 years. And they know this because you can mark an outbreak, and then those people swore off meat after that. Once they survived this infection, they said, I'm not going to eat any more meat. And they said, by the way, when you die, do you mind if I take a piece of your muscle tissue to see if you've got any larvae? I'm sure it wasn't said like that, but they had a lot of follow-up, which has accumulated this data that suggests that some of those nerve cells will stay alive. Now, what are the factors determining that? So I did some, I thought, cutting edge research. It involved a microtome, by the way, which is a cutting edge.
That was the joke, the cutting microtome edge.
It was right. That's right. And what I was trying to do was, what happens inside of this altered portion of striated muscle is that any nuclei from the host that gets trapped within this system, they divide. So you get a doubling of host genome within the affected area of the muscle cell. And sometimes it's like 10 nuclei, sometimes it's 20 nuclei. I've had nerve cells as high as 40 nuclei from the host that are all doing the same thing for this parasite. And I speculated, although without any proof whatsoever, that the nerve cells that live the longest are the ones with the most nuclei. It makes sense, at least, to know that the nerve cell is churning out message, which is telling the host what to do in order to keep this parasite from dying. And part of that, of course, is this circulation that is elicited. It's not an ordinary circulation. These are not capillaries, all right? These are vessels which are found in exocrine glands, sinusoids, they're called sinusoids. The liver is famous for its sinusoids. The adrenal glands are famous for their sinusoids. Well, Trichinella is also famous for its sinusoids. They're leaky vessels. They don't carry oxygenated blood, which is very important for this parasite because it's an obligate anaerobe from the start. So I know that one of the papers we're going to review suggests that it's an anaerobe because it can survive in a carcass after the animal dies. But in fact, it's always been an anaerobe. It starts out as an anaerobe. Remarkably, its mother was an aerobe. So mama has good Christie in her mitochondria. She has no stored glycogen whatsoever. She's totally dependent on the host for the energy sources that she utilizes to make the newborns. But once the larva is inside the muscle cell, it shuts down all of that aerobic metabolism of a skeletal muscle cell, and it decouples the mitochondrial activity that produces ATP. And you can see this on an electron micrograph. It's quite easy to see the inner matrix of the mitochondria is totally missing. If you go just outside the nerve cell into normal tissue, unaltered tissue, the mitochondria are perfect. So the worm is doing something to make sure that this unit remains anaerobic at all times. And its metabolism bears this out. I did some studies as a graduate student. Our department obtained a brand new respirometer. And of course, I wanted to use it. I wanted to just learn how to use one. And it's a machine with lots of little knobs that you keep turning to equilibrate when the living material inside the chamber either produces CO2 or does not produce CO2. And you end up with something called the respiratory quotient. And the respiratory quotient, if it's 1 or more, usually 1 is a good number, it consumes glucose and secretes CO2. So you're constantly adjusting to keep up with the metabolism of an aerobic cell. With Trichinella, they don't secrete CO2. They secrete acids of various kinds as a result of lipid breakdown. And they also secrete pyruvate, but they do not secrete CO2. And they are 20% glycogen. So if you mashed a bunch of larvae up and separated out all the components, 20% of the dry weight of that worm is glycogen, and that's its reserve energy source. If you do the same thing to an adult female, no glycogen, none. She's a strict aerobe, a microaerophilic aerobe, whereas the larvae is an anaerobe. And I'm making that distinction between the two because one is very short-lived. The aerobe burns its candle at both ends, produces larvae from day 5 until, say, day 10. At that point, the immune system kicks in and starts to inhibit her ability to produce larvae. Doesn't kick her out, just absolutely stuns her in terms of her nutrition. And that's the basis for immunity. Eventually, they are kicked out. But in the early stages of this infection, the enteral stage lasts for about 2 weeks, maybe 3, and then it's gone. The parenteral stage can last 50 years if it's in a long-lived animal. So that's the story. And Vincent, you're absolutely right on the money saying that the host that harbors the larvae in the muscle tissue is not the final stage of this life cycle. So it's an incomplete life cycle. It requires 2 mammals in order to complete it. But any mammal, that's the point. It's a generalist in that sense. It can infect virtually any carnivore everywhere. So there's a parasite named Trichinella nelsoni. I knew Nelson. He was a wonderful parasitologist. I worked at Liverpool and had some remarkable stories, most of which I cannot tell on the air, but I'd be happy to sit with a glass of scotch with you and tell you all about
let me
how.
ask you, Dickson.
Yeah, please ask me something.
So we're gonna learn about the problems with nativa, for instance. Now, Trichinella spiralis versus nativa. I would be more worried if nativa got into our pig population. Do you think it's just happens that it has not gotten into there, or is there some sort of a preferred tropism? Do certain subspecies do better in certain animals? Do we know?
Sure, let's talk about that. If you're interested in the epidemiology of how Trichinella gets to humans, in the old days, let's say before 1950, an autopsy in the United States, when we used to do them, the rate of Trichinella infection in some places was over 40%. They could find larvae in the diaphragm tissue of 40% of the cadavers that came to autopsy. And all of that started to change around the early 1950s through the 1950s to 1960s, sort of an awareness of public health and meat safety and animal husbandry. And it's an extremely easy infection to control. All you have to do is prevent rats from taking up residence on a pig farm, because the pigs will instantly eat those rats. They like a moving target. They kind of have fun chasing them down and consuming them. And the pig farmers in the old days not wanting to waste anything, when they had their pigs slaughtered, they saved all the organs that were not sellable, and they brought them back to the farm and fed them to the pigs. And now you know how that happened. Now, I'm blocking on the name of this publication, but it was famous back in the 50s. A series of articles were written for the New Yorker magazine. And Daniel, perhaps you remember the name of the author who became famous for writing these medical mysteries. And one of them was called A Pig from Weehawken. And Weehawken is in New Jersey.
I'm trying to think if it was Lewis. Is that correct?
No. He was too high up on the food chain of authors. Yeah. This guy he had a French name, as I recall. I'll get it for us. And he wrote the story about how Trichinella figured in the New York restaurant industry. The pigs from Weehawken were butchered and shipped to New York, which is just across the Hudson River. The scraps from all of the restaurants were shipped back to Weehawken. So if the pig happened to have Trichinella, and no one inspected for this, of course, that's not true in Europe, but it was true here. Nobody inspected for this because the USDA, in all of its wisdom, once said, well, if we inspected for it, then what do you expect us to do?
You would find it, and then what would we then have to do?
That's
I feel
right.
like I can think of so many parallels.
Well, it's not unheard of for any infectious agent that you can inspect for. So then they would say, well, sure, if we worked it out for Trichinella and we can inspect for it, you know how many other infections there are that we could inspect for also? Toxoplasma, neurocysticercosis. They do inspect for cysticercosis, but they make a cut down the side cheek of an animal and spread it open. And if they don't see it, they declare it negative, which is ridiculous. It could be anywhere. So the point is that it costs money to somebody once you find a positive animal. And they were unwilling to subject the pig farmers to an added expense.
I remember growing up, and I don't know if this is as common today, but the one thing that was drilled into me as a child, and maybe there was only one thing, that's how it turned out this way, which is that you do not eat undercooked pork.
That's right. That's exactly right. Medium rare pork does not exist. It does now. And it does for a very good reason, and that is that we haven't had a major outbreak of Trichinella due to an infected commercially raised pig in 20 years. So we've done a good job of epidemiologically controlling this infection. In Europe, they still inspect. There have been remarkable case histories of an inspector who either fell asleep and missed the positive that came up on the screen because they had this device called a trichinelloscope. They would take an enormous circle of diaphragm tissue from a pig and put it between two plexiglass plates and then apply pressure, and it would spread out, and then they would shine a very strong light through it onto a screen, and you can see every larva at that point.
How could a person fall asleep when their job was that interesting?
Yeah, well, when you've got 300 pigs to go through a day, and you get to day number 12, and one of them was discovered because he saved all the diaphragm tissues and brought them home and fed them to his dogs.
Yeah.
And his dogs died.
Oh, my gosh.
And he died from a piece of meat that was infected that he didn't see. And that epidemic was, he was fired, of course. At any rate, there's a lot of rich history to this infection.
So I love the, and I think here there really is this great connection of understanding the life cycle and understanding how one presents clinically with this disease.
Oh, that's right. Okay, so so we had a very nice graph. I'm wondering, Vincent.
Figure 21.10 for those of us with photographic memories.
If we had a textbook handy, we could turn to it because it's in our textbook.
On page 247, but I could walk you through that.
Well, since you're the clinician in residence, take it, Daniel.
Well, it will be good because then you're going to walk us through an outbreak and we'll see were we right?
That's right.
That's right. Wrong.
Yeah, go ahead.
And so it's a great figure. It's going to be in our edition 8, which is coming out, I hope, before Christmas, Dickson. It's going to be on you and me because Chuck Knersch and I are almost at the point where a PDF will be presented for the two of us to read to one another before the final printing. But
if you talk about going to sleep, that's the time to do it.
Maybe we'll make an audio book. But so you can almost think about what the manifestations should be based upon this wonderful life cycle that Dickson that you just shared with us. So initially, where do you think the symptoms are going to be? Are they going to be muscle pain or are they going to be GI?
No. I think, I already know the answer. So
it's always the last answer. I make it easy. But, because the first stage is going to start in the gut, right? So there's a short incubation period. It ranges a little, but it's usually less than a week from the time the food is ingested. Some people may start having symptoms within a day or two.
That's right.
And the first manifestation is going to be GI. So there'll be some GI upset, some nausea, vomiting, diarrhea.
Stop right there. I want to add a note here.
Yes.
Because what you get in the gut tract during the early phase of the infection is hypersecretion of the gut. And fluid fills the gut. And in mice, this is quite evident. And then you get a weight loss as a result of that. But, so what's the purpose of that, do you think? What purpose does that serve? The parasite.
To flush out the parasite?
No. The gut tract stops moving. It becomes hyper secretive. Mm-hmm. I knew someone who worked on that aspect for 20 years. All of the mechanisms involved in we even lose our wheat germ agglutinin receptors for all of the columnar cells as a result of that infection. But there's a purpose which allows the parasite the luxury of actually, after having developed to an adult, which is only 36 hours into the infection, of coming out into the milieu of this fluid and finding a mate. Now, one of the most fascinating aspects to this is the ratio between males and females at 36 hours. You give 100 larvae orally to a mouse, assume they're all infectious.
Which you did.
Which I did many times. And if you take the worms out at 36 hours and separate them by sex, how many worms do you think will be female and how many worms do you think will be male? Ordinarily, you'd say 50-50, right?
I'm thinking that women are more important just as a general rule in life.
You're not wrong on this one. You are not wrong. You would get between 60 and 70 females and only 20 to 30 males. Now, here's the mystery. If you
go to math on that, 10 or non, it's 10 or modern. They're non-binary.
No. Here's where we started our research on where does mating occur and how does it occur? Because at 48 hours, which is only 2 days late, 2 days after infection, you can take all of the females and put them under the microscope, and you can see that every single female is inseminated. Every single female. So within a very short period of time, between 36 and 48 hours, the males and females get together, and it is like a Hugh Hefner orgy.
And Vincent, thus why we have such high rates of syphilis.
Trichinella doesn't use any protection whatsoever. So what we ended up doing was looking in the fluid for the worms, and we found some, but we didn't find all of them. And it was disappointing because the fluid was there, it was clear to collect, easy to get, and we looked at 2-hour intervals from 30 hours out to 40 hours. And we still didn't find the moment when raises another question, of course.
Yeah.
If it happens that quickly, how does a male worm know not to mate with a female worm that's already mated?
Did she just ask, sir?
Yeah.
They're very polite worms. And there's excuse me, is this Dan's taken? Well, that's another question we wanted to know about, because this happens incredibly fast. This is a worm not just in a hurry. This is a worm that's really in a hurry.
Mm-hmm.
So its life cycle is truncated in the gut, and it's expanded in the muscle cell. But you're back to the clinical symptoms.
So let's keep marching through our life cycle. So now we've got this gut phase, right? It has yet to become invasive, and as such, secretory diarrhea, nausea, vomiting,
right?
But no fever yet. No eosinophilia yet.
So what's your diagnosis, doctor, at this point? What are you thinking?
Think about it. Someone shows up, they come into an ER, they say, it doesn't feel right. I don't know. Maybe I ate something. I'm nauseated. We would just say, you got a case of gastroenteritis. You're not running with fever.
Food poisoning.
Food poisoning, yes. And we would send them away unless there was an ID doc who maybe was paying attention, or an ER doc who had maybe seen several people and said,
yes.
Did you also go to that event?
That barbecue in the outdoors, that's right.
A little bit more history. But so then, after the first phase, then you start to get the penetration, you start to get the fever, you start to get the eosinophilia, you start to get the whole inflammatory migratory process. That's when the CPK goes up. That's actually the stage when we start to see people succumb to this infection. And as I mentioned, most people do not succumb, as we'll see. But maybe that's enough for us then to get into some of these papers.
I have one more question.
Oh, yes.
No, go ahead.
So one of the symptoms is this ocular swelling, right?
Periorbital edema, that's right.
Well, what is that from?
Bilateral periorbital edema.
Yeah, it's actually, so it's periorbital edema. So it's really, the whole person becomes swollen, including swelling around the eyes, swelling in areas that are not gravity dependent. And so they'll actually even get this conjunctivitis and subconjunctival effusion and swelling, or chemosis. And so what you're getting here is all of this leakiness of the vessel. So let's say you had heart failure, right? Your swelling will often be dependent, swelling in the legs. But when you have either a lack of protein or an increased permeability to your vessels, you end up with gravity-independent swelling. And that's what we're seeing. We're seeing,
and that's caused by the larvae penetrating out of the vessels into the muscle.
Yeah, and this is probably something that benefits the Trichinella.
Well, I think it's something that they can't help. Mm-hmm. In order to get out of this circulation and into a cell, you've got to break the circulation. And when that happens, you get this little petechia. You can actually see them under the fingernails much better.
Yep.
So
wait,
I wanted to pause here.
Outbreak, right, Dickson? An outbreak of human trichinellosis?
Yeah. I wanted to pause for a moment, though, and ask Dr. Griffin whether or not he can surmise, based on what he just said, which stage of the infection causes the eosinophilia.
So I think that it's not going to be while it's still in the gut. You're not going to see any eosinophilia until the larvae are penetrating and moving through tissue. Now, I think that's a great answer.
Rule. I love that answer, because that's what I thought too. So we can actually explore that experimentally, and I did. We can collect adult females at day 8 or 9 when they're busy shedding larvae, and we can collect them in vitro. Then we can screen out the adult worms, So now we've got just newborn larvae. And then we can concentrate them. And then we can, with a 26 gauge needle, we can actually inject them intravenously. And we did that in mice.
Interesting.
And so your prediction was that that's the stage that induces the eosinophilia.
Well, I'll
No,
even,
no hedging, no hedging.
No, but I'm
sorry, you've already put the X down in the box.
That is when the eosinophilia does start, but it's not when it peaks.
It is. So let's fast forward now to the end of that experiment, which is 30 days. We leave 30 days for the nurse cells to fully develop. Okay, so now they're all fully developed. We've taken blood every single day. We didn't find a single eosinophil.
Interesting.
Not one. Not so good, right? Okay, so let's, what else can we control to see if it's that stage or not? So there's a drug called metriphenate. And metriphenate actually cures the infection in the gut. Whatever time you want to give the drug, the parasites are kicked out at that point. So let's do the same experiment, infect orally this time with larvae. And within 1 day, they will transform into adults. And we'll use metriphenate at day 1 and get rid of the infection altogether. They've been exposed to the gut, but they haven't developed to adults that are reproductive yet.
Yeah, so no
we got no eosinophilia. We got none.
Yeah.
Okay, then we went to 2 days. We got none. We went to 3 days, and we got a blip. Still, they're not producing newborns. We went to 4 days. We got a larger blip of eosinophilia. They're not producing larvae. They've been adults now for 4 days in the gut tract. And it turns out that the enteral stage of the infection is the inducer of eosinophilia. And that was a complete surprise to us because we thought, of course, it was going to be a systemic infection that induced this generalized malaise, which resulted in the pathology that we see in the infection. But in fact, the adult worm induces eosinophilia. And what's even more surprising is that it's an essential component in nerve cell formation.
Mm-hmm.
Without eosinophilia, well, no, I will back off that remark because I didn't discover that. Somebody else did.
But that is an issue. If you can somehow decrease the eosinophils, you can decrease the nerve cell formation.
Well, you'd think so, wouldn't you? But that experiment that I told you about where we injected newborn larvae and got nerve cells without the enteral phase.
And there's no eosinophils there. That's a little
none.
Odd.
And we still got perfectly looking or perfectly looking nerve cells. So I disagree with that finding. And I had a raging argument with a very good friend of mine who worked at Cornell University.
This is what parasitologists fight about, Vincent.
Well, yeah, no, virologists do too. Come on, you're going to fight about anything. But
this is interesting because clinically we have this idea that if we can somehow blunt the eosinophilia, and maybe we'll even use steroids, that we can somehow prevent the number of nerve cells. But
you do use steroids, and in fact, that is one of the treatments for acute trichinella. Is to use steroids to try to prevent innocent bystander effects. And it works, but it prolongs the life of the gut tract infection, and they produce more newborns as a result. So the woman who discovered, or at least published, the paper that said eosinophils are absolutely required for nerve cell development, her name was Judy Appleton. And des pommiers means apple trees. So there was an apple tree fighting one of the products from the growth of the tree itself. We never did resolve it. And it might be a strain difference of animals. There's a lot of reasons why we got differences in our results. But newborn larvae that resulted in nerve cells never induced a circulating eosinophilia.
Interesting.
Dickson, tell us about this outbreak of trichinellosis.
Well, I think Daniel should start, and then I'll pick it up.
Sure.
The cases were first.
Yeah. So, because we're also going to talk about what is that? Climate change's impact on Trichinella.
Yes.
But let's talk about this. So, this is very appropriate. We had a recent outbreak here, which we were actually intimately involved in. But there's this outbreak report of human trichinellosis, Arizona, Minnesota, South Dakota, 2022.
Right.
And this is an outbreak report published in the CCDR, May 2024. And we have a list of authors, Shama Cash-Goldwasser, Dustin Ortban, Muthu Narayan, Connor Fitzgerald, Keela Maldonado, James Curie, Anne Straley, Sarah Sapp, Henry Bishop, Billy Watson, Margaret Nedja, Yvonne Kvorstrom, David Berman, Sarah Park, Kirk Smith, and last author, Stacy Holzbauer.
Sounds like a physics paper.
Well, they've got 10 different affiliations, right? So we've got the CDC EIS, the CDC Epidemic Intelligence Service.
Right.
And that's actually Shama Cash-Goldwasser, South Dakota Department of Health, University of Minnesota, Arizona Department of Health Services, Maricopa County Department of Public Health, the Lakeview Clinic, Division of Parasitic Diseases and Malaria, Global Health Center, CDC, Medical Affairs, Carius Incorporated, Minnesota Department of Health, and the Division of State and Local Readiness, Center for Preparedness and Response, CDC. So we've got a number of folks here involved, and it starts off in July of 2022 when the Minnesota Department of Health is notified of a 29-year-old man. You're going to have to start thinking about what stage of disease, 29-year-old man hospitalized with fever, severe myalgias, periorbital edema, eosinophilia, and some other laboratory abnormalities. So what are we thinking? So we're thinking not the first week. We're going to have to ask him about that. We're thinking this is sort of probably about third week or so.
Yeah, that sounds right.
He's got the eosinophilia. He's got that non-gravity edema, the periorbital edema. He's got all the muscle aches. And then they tell us, so he's been hospitalized like several times, right? So he initially seeks care in July 4 times, ends up hospitalized twice over a 17-day period. During his second hospitalization, they must call the ID doc, who asks him, tell me a little bit more. It is, I gotta say, like a step on my soapbox. It is amazing how much money gets spent in medicine before someone just says, hey, let's send in someone to get a good history.
Right. Exactly right.
So tell me a little bit. You sit down. I love to do this. You sit in the chair, you start chatting. Next thing you know, you hear about this history of bear meat consumption. And this leads them to ultimately get the diagnosis. But then we go back in history. So now we really get the history. And this should have happened, 6 days before the symptom onset. So less than a week, 6 days, he and 8 other family members from 3 states, right? Everyone's getting together, right? They gather in South Dakota and they share a meal that included bear kebabs, right? So this is chunks of bear meat on a stick. And this had been harvested by one of the family members who had been hunting up in Canada. Now, they take the meat and they do...
Wait, how old was the meat?
Well, so they gather the meat and it had been in a freezer. Are you ready for it? 45 days. So it's been frozen. It should be fine.
You betcha.
Freezing should kill this, don't you think?
It.
They thawed and they grill it with the vegetables. But did they cook it enough? No. It was inadvertently served rare.
Right.
How do we know that? Well, we don't really know this because I don't know how many of our listeners have eaten bear meat, but bear meat tends to be dark in color. So the only way you're going to know if your bear meat is cooked enough is to actually stick a thermometer in it because, you're normally thinking, venison, maybe elk, maybe beef. You can look at the color and that's going to tell you how well it's cooked. But bear, you really can't just look at the color and know how well it's done. So they end up serving it rare and end up with a number of folks getting sick.
So interestingly enough, to add to this story, is the fact that what do bears do, not in the woods, which is a typical question, but what do bears do in the winter?
They hibernate.
Yes, they do.
They also shit in the woods, Dickson.
They do. Not in the winter.
It's kind of amazing, right? 6 months of just, boy.
Of nothing. And if you're a mother bear, you give birth and the babies are born and they nurse and the mother is still asleep. Totally asleep throughout the whole event, right? So the point is that hibernating animals have special proteins in their blood to prevent their muscle tissue from freezing. It's an antifreeze molecule, which is common throughout nature. And fish have a similar molecule. In fact, there are numbers of wonderful studies of fish that have been harvested from the Antarctic Ocean, the Southern Ocean. And they live essentially in a minus 3-degree water temperature, but it's hypersaline, so it doesn't freeze. But nonetheless, if you've got just water in your cells, you're going to freeze. But these fish are happy as, dare I say, happy as clams, because of the molecule that they've got circulating in their blood, which prevents them from freezing. And by the way, those fish don't have any red cells.
Dickson, if you get trichinella once, can you get it again?
Great question. Trichinellosis is a disease.
Yep.
The answer is no. You can get an infection with trichinella, but you will not become sick from the infection.
Because I noticed one of these people didn't get sick, even though he ate. So I wonder if he already had it.
Well, yeah, maybe that was the hunter. Right? Maybe. So when you hunt a bear, I mean, I don't care how small they are, they're still well over 200 or 300 pounds of meat.
They're big. Yeah.
And they're enormous, and they're scavengers. All right. They eat carrion. They eat whatever's there. In fact, they will eat vegetables if that's all that's available. So Dickson, bears are basically omnivores.
Has anyone done a survey of bears and see what fraction are trichinella positive?
Yes, they have. And it's quite easy to do the survey. You can either look in the stool for the DNA of the organism, which is the easy way of doing it, because bears do shit in the woods. And the DNA of their inhabitants in their muscle tissue comes out as well. Or you could do a hunter survey, because every bear that's shot has to be registered with the Fish and Game Department.
Where does the bear get the trichinella?
How did it get it?
Yeah, from other animals?
Or from rummaging around in the garbage, or, that sort of thing.
But so bears are omnivores, right?
They are true omnivores. That's right. Remember, Kathy Spindler on our show This Week in Virology took a trip to Glacier National Park, and she was at the 8,000-foot mark, camping out. And she said she looked out of her tent, and she saw a mother grizzly bear and babies turning over rocks.
Yeah.
And she said, what were they doing? And I said, they were eating moths. She said, how did you know that? I said, well, I saw a special on television about it. But the point is, that's how far into the wilderness ecological setting bears will go in order to get enough protein. They have to eat like 20,000 moths a day. And the mother bear teaches the babies.
Remember was it Alan's pick or Rich's pick, the fat bears of Katmai?
Yeah.
Oh, yeah.
Which, you can watch the bears of Katmai. So you go to Homer, Alaska, you continue west across the water, Katmai, where some of the fattest grizzlies on the planet.
Exactly.
And they're fattening up for winter, and they are just harvesting salmon over there.
You bet. And in fact, when they do, they don't eat the meat of the salmon. They just strip off the skin and eat it, because all the fat's just underneath the skin. And so those carcasses that they create form the basis for a fertilizer for the plant life that grows along the banks of the river. Without that, there would not be the ecology that you have today. So dead salmon that are created by bears are very important. Anyway, back to Trichinella.
Yeah, but I just want to wrap up the this thing. So this is another one of those areas of exciting research that we got to get together a parasite meeting to fight about this, Dickson. But is it the Trichinella species or is it the host? And I'm going to say that there's some evidence suggesting both might be involved. I sort of dove into this when there was a recent outbreak, and I was communicating with the folks down at the CDC. We covered a little bit, but yeah, and that was sort of why I brought up Nativa versus spiralis. Like, what if Nativa got into pork? Would we have more of an issue? Would it be more freeze resistant? It looks like it's both. Both are probably contributing.
It's had lots of opportunities, and it hasn't so far. But that doesn't say it couldn't happen. Oh, I will totally kill the epidemiological story that I like the most is a farmer story, where the CDC is convinced that this farm is the source of the infection, but there's no evidence for it because the farmer is meticulous with the scraps of pork. And he buries them, and he just gets rid of them altogether. So this was told to me by an EIS officer, and he said, and he was looking around, and he got out to the barn, and on the barn were several pelts, and one of them happened to be a fox pelt. He says, "Oh, I see you're a trapper." He says, "Oh, yeah, we do a lot of trapping." Oh, that's interesting. He says, "Well, what do you do with the carcasses of the animals after you've skinned them?" He says, "Oh, we grind them up and feed them to the pigs." So foxes are equivalent to small bears in terms of their eating habits. And true, they will eat some vegetable material, but they're probably more carnivores than anything else. So let's now look through the telescope rather than through the microscope and just briefly discuss
we're going to discuss climate change.
Yeah, let's discuss whether or not it's a real phenomenon. Climate change is certainly a real phenomenon, but whether it's interrupting the scavenging habits of animals or whether it's all speculation.
Let me ask Daniel just one question.
Yeah.
They treated this patient with albendazole. Yes. Is that typical? And will that get rid of all the worms?
So,
great question.
So it is typical. I'm going to give you the typical. It is typical when you're considering the diagnosis to use, albendazole or mebendazole. The other is if you're really confident, you might step in with steroids as well. So you might, particularly at this stage, early on, you're sort of a little hesitant with steroids until you're certain of the diagnosis. But no, the standard would be to use albendazole and a glucocorticoid.
Well, what about thiabendazole? That's an older drug, but it's shown to be very effective against the first 2 weeks of the infection.
Oh, yeah. I think whether you use, so thiabendazole we don't tend to use very much, but mebendazole and albendazole tend to replace it. But yeah, it would be fine. It'd be the same.
You don't think about Trichinella until it's too late. Now, will those 2 drugs or 3 drugs wipe out the muscle phase? And the answer is no.
No. Once you've got nerve cells, right? I mean, you're not.
That's right.
Okay.
They're impervious. So Eduardo Pozio is a friend of mine, and he wrote that paper that you picked for climate change and whether or not we've got a problem.
Is this one of the few things where climate change is going to help us out? Maybe a little warmer, a little longer summer, a little less Trichinella. I mean,
well. You can say that, but the studies that so far are valid for this, where they've taken carcasses that they create in the lab, and you put them either in the refrigerator or you put them under the fume hood at room temperature, and you sample them every day, and you digest the muscle tissue with pepsin and hydrochloric acid, and then you infect animals with those to see if they're infectious. And the the wisdom is that most carcasses will produce an infection for the first 2 weeks after the animal dies, and then after that, it's not really a factor in terms of transmission. Less than 2% of the larvae are still viable, and Eduardo claims that Trichinella was selected for anaerobic life to survive in carcasses after the death of the animal. And I claim that it was selected for life in nurse cells, which precedes the death of the animal by some 20 or 40 years in some cases. And that has nothing to do with whether it can survive in a carcass afterwards or not. Because in nature, I witnessed a kill, for instance, when I was on safari with my wife in Zimbabwe. Not Zimbabwe. In Botswana. We saw a cheetah actually take down a male antelope.
Wow.
And it was happening right in front of us. And I got a whole series of wonderful pictures of this. And the cheetah stood guard over this carcass and wouldn't take a bite. And the guide said, this cheetah will not take a bite of that carcass until it's sure that the commotion caused by the killing of the animal didn't attract other animals like hyenas or lions or even other cheetahs. So we sat there for an hour watching this cheetah move the carcass from here to there, sort of stood up and was very proud of the fact that it had killed something and it was going to be able to eat really well. But we couldn't tell whether it did or not until we came back the next morning. The carcass was gone. It was gone. Now, I don't think the cheetah actually ate all of that. I think the cheetah probably was scared off by some pack of hyenas, and they ate everything, including the bones. So they leave no evidence whatsoever.
Yeah, the hyenas will do that. They'll eat the bones.
They eat the bones, and they're the only ones that can do it. But carcasses don't last more than, let's say, 2 or 3 days.
So this whole thing of like 2 weeks survival, it may not really be.
It doesn't. I don't think it really matters much. And in particular in the Arctic, even though the temperatures are changing, they haven't changed that much, and there's still a lot of ice there in the winter, and there's still a lot of cold. And so Nativa does very well under those conditions. And in fact, I wouldn't be surprised if someone actually dug up a carnivore like a saber-toothed tiger or a sloth and looked in the muscle tissue of the sloth and found living larvae. That would be quite a find, but it would certainly illustrate the power of
it would be interesting how far. I mean, I know we've done it. What have we done in the lab? Like 5 years in grizzlies, but who knows what the actual limit is?
Right. But I think the big worry is the first 2 weeks. And the carcass will be discovered by smell.
Yeah.
And the game is over at that point.
Cool.
So I think Eduardo wrote a good article. I enjoyed reading it. And he even used one of my references.
All right.
But the idea is that climate change is going to increase temperatures. It's going to be less snow coverage, and it's going to reduce transmission of Trichinella, right?
No, that I think it would actually increase transmission.
Yeah?
Yeah, because if you don't have any snow, the carcass is always available. It's not buried underneath a heap of ice. So, yeah, animals are always hungry. They never pass up a meal. Even if it's a rotting carcass, they don't pass that up. Which I can't figure out how they do that, because you and I wouldn't eat that stuff on a bet.
Yeah.
I mean, even if it was non-toxic, you're looking at a piece of meat that's probably autolyzed and digested already, and you're just going to drink it almost. It's just, and animals just dig into that stuff like it was dessert.
I'm thinking of the buried shark that I once ate in Iceland, where you bury a chunk of shark and you let it ferment in the ground for a year, and then you regret doing that.
Is that there's a Norwegian dish I forget the name of this dish. It's fermented shark. It is fermented shark.
Yeah.
And no, thank you.
Not my favorite.
Not for me, thanks. Even with a scotch.
All right.
So Dickson, if the biomass of Trichinella declined, would that be a bad thing?
Not in my view. No, but I don't think it will. I honestly, I can't imagine it. I think when the temperature goes up, you get more animals. You don't get fewer animals. You get more animals. The mating season is longer, that sort of thing.
Okay. That'll do it for TWiP 246.
What a wonderful TWiP it was, too.
It's, actually, we have one letter. Let's read it.
Sure.
Maybe you should read that, Daniel. Read that.
Go ahead.
Frank writes, I'm a loyal listener and sometimes donor to Twix for 18 years. A grand thank you to Vincent and all participants for the Twix education and entertainment. Each time I hear you discuss Lyme and other tick-borne diseases, I hope to hear detail about the often referred to minimum attachment time to transmit. And we've got the CDC link here, where it said ticks needs to be attached for more than 24 hours and begin filling with blood before they can spread Lyme disease bacteria. If true, this should provide plenty of time for the average person and their partners to look for and find an attached tick. Living and hiking in Northwest Connecticut for many years, I'm very familiar with the routine. Who better than the TWiP crew to discuss the step-by-step process of various ticks attaching, injecting their chemical cocktails, and drawing blood? Dickson and Daniel often provide this in great detail for reduvid, tsetse, or mosquito disease transfers. Just as common sense is surprisingly uncommon, scientific communication is often frighteningly unscientific. Thank you all for being the source for factual, no-nonsense, and scientific approach to presenting information. Best regards, Frank. So I will say this is interesting. So this is true. It probably takes more than 24 hours for the deer tick to transmit the Lyme, the Borrelia spirochete, which is, thus the songs about, checking my partner for ticks, the famous Western song, and something that you should do. But I think there's a couple issues here. One is there are other pathogens in the ticks, which actually can spread in as little as 15 minutes. Can you believe that? And also, when the lymphal tick first attaches, it's so small, it's the size of a poppy seed. Right. And often it's up in an area where maybe it would be indiscreet for you to be checking unless you were very intimate with the partner.
Right.
All right, that's TWiP 246. Show notes, microbe.tv/TWiP. You can send us your questions or comments to twip@microbe.tv. And again, if you like this work, this science communication, please support us, microbe.tv/contribute. Daniel Griffin, Columbia University Irving Medical Center, parasiteswithoutborders.com. Thank you, Daniel.
Thank you. And everyone, be safe.
Dickson Despommier is at trichinella.org, thelivingriver.org. Thank you, Dickson.
Welcome, Vincent.
I like to hear you share your knowledge of so many years with us.
I enjoy being asked. I do.
I'm Vincent Racaniello. You can find me at microbe.tv. Music on TWiP is by Ronald Jenkins. You've been listening to This Week in Parasitism. Thanks for joining us. We'll be back soon.
Another TWiP is parasitic.
What did this mean, Dickson?
I wanted you to say it.
I thought you meant yourself.
No. I was pointing to you.
It's okay. It's fine.